Oh yeah, sure. Thank you. No problem. Yeah, I'm gonna just go now. Oh, it's over. Thank you.
The meme was the same. Or no, the meme was the meme was the same. So what it means is
Okay, it's movie time. Yay, okay. So, the cool thing about mitosis is, unlike signaling and unlike protein trafficking and a lot of the other things that we've talked about, mitosis is really visual, okay? So it's one of those things that happens on such a large scale, and all of the components in the cell that are involved are so big. Chromosomes, the mitotic spindle, everything is so large, you can just look at it and you can see what's happening. So that actually makes it relatively straightforward to go through and understand, because you can actually just look at each event that's going on and go, so that's what's going on. I mean, you can actually see the mitotic spindle form. You can see chromosomes being captured, lined up. You can see the events during anaphase where the chromosomes move and the poles move. So it's really nice and visual. So all you really have to do to understand it is, well, what's going on to make all this occur, okay? And it's complicated, but not that. What doesn't make it complicated, what makes it complicated? Sorry, rewind. What makes it complicated is a lot of things happening at the same time, but they are all separable biochemically, okay? So you can go through and you can analyze each of these events. It's sort of like a ballet being choreographed. You can go through and you can just focus on each individual dancer and then see how all of this stuff is playing out together, okay? So this is just showing you a couple of embryos, sea urchins, Drosophila. They're expressing GFP tag beta tubulin, so you're actually looking at the spindle itself, and it's really cool to be able to see, okay? Now, here's what I want you guys to be looking for, all right? This is an image that's now focusing on the chromosomes, and this is kind of taking you through the process from start to finish, and this is pretty much how we're going to cover it. So the first thing that you're looking for is we have to get rid of the nucleus. With the exception of Saccharomyces cerevisiae, which forms a mitotic spindle inside the nucleus, all real eukaryotes form a mitotic spindle from the outside, in the cytoplasm, which means that we've got this barrier called the nuclear membrane between the microtubules and the chromosomes that they have to capture. So the only way that that works is we get rid of the nuclear membrane every single time our cells undergo mitosis. So that's going to prove to be one of the targets of cyclin CDKs. Then we got to form the spindle poles. Then we have to get all the chromosomes once we get them in. This is what's happening right there in the video. You're having to get all of the chromosomes lined up in the middle or the equator of the mitotic spindle, because that's where the plane of cell division is going to go right through there. It's going to bifurcate the cell right through there. And you can also see this is why anaphase goes really fast, because the chromosomes have to get out of town before the plane of cell division comes right through there. Okay? But one of the things that you notice is the cells don't go into anaphase until they're good and ready. And you're going to see that there's a checkpoint there that is going to stop the cell from going into anaphase until all of the chromosomes have been what's called captured. And that is, they are all attached to the mitotic spindle. And in fact, that all the chromosomes have organized in what are called bipolar or amphittelic attachments, meaning each sister chromatid on a chromosome, after it's undergone DNA replication, each one of those duplicated chromosomes has to attach to the opposite mitotic spindle. So that way the two chromosomes, each daughter cell, gets a chromosome. If that doesn't happen, we get chromosome missegregation. That means one of the two daughter cells is going to miss that chromosome entirely. The other daughter cell is going to have an extra copy. Neither of those are good. And in fact, it's a common mechanism for how tumor suppressor genes are lost in cancer cells that we'll be getting into. Okay, so we go, you can see right here, here's the guy who's holding the whole thing up. This chromosome takes a while to get attached, then he finally goes in, boom, they're gone. Then what we have to do, as you can see, cytokinesis goes really fast right after that. And then we have to reform the nuclear membrane, and then we have to take our chromosomes, which are enormously compacted, into these little things called mitotic figures, so that they're little compact bundles that can be segregated by the spindle, but they can't do anything. They're inert as far as transcription goes. So in order for the cell to go back into interphase and start expressing genes again, all these mitotic chromosomes have to decondense and go back to their interphase structure. And here's the mitotic spindle checkpoint. Where you see this guy here, he just can't get it right. And everybody else is waiting. Everybody else is hooked up, they're ready to go. But if one single chromosome isn't attached, it keeps the entire process from happening. And not surprisingly, what's driving all of this? Kinases and phosphatases. Okay. Oops, sorry about that. Okay. The way to look at mitosis is, it's a series of sequential events that we can divide into early events, or early steps, or late steps. Early events are driven by MPF. Think of it as the kinase part of the program. And what that does is that part of the program gets you from interphase all the way up to metaphase. So think of metaphase as the culmination of the MPF part of mitosis. It gets you to that point. When we now go from metaphase through into anaphase, telophase, cytokinesis, those are the late events. And that's where we see a switch in the program. Instead of the program here being driven by protein phosphorylation, here the process is being driven because MPF has been inactivated, and that's because it's handed over to the anaphase promoting complex. So this is where the APC got its name. So in essence, the way to think about it is, phosphorylation gets you up to metaphase, and to go from metaphase through the rest of mitosis requires dephosphorylation, and it's going to require ubiquitination and degradation. Those two things are going to drive the second half of mitosis. So it's really just a binary system, okay? All right. So here's how we can look at the two functions here. MPF and anaphase promoting complex, okay? So these are some experiments that are being done where all we're doing is that we're taking a look at the chromosomes up here as they go through mitosis, and we can do that by staining DNA with a fluorescent dye called DAPI, okay? So if you take a look at the top, here's the time frame. You can see it's pretty quick, 40 minutes from metaphase. Here we're in early anaphase, the chromosomes are starting to separate. Here they're nicely separated. And you notice the fluorescent staining there is very intense. And that's because the chromosomes are all condensed. They're all really packed together. Notice when we come out on the other side here, after anaphase, and now we're in telophase, you notice that the fluorescent staining of the DNA is starting to become more diffuse. And that's because the chromosomes are decondensing. They're going back to their interphase state. So what you're looking at in these panels is the top graph, or the top panels, are showing you the chromosomes. The bottom panel is that we're staining with a fluorescent antibody against tubulin. Okay? So here we're following the spindle. And the one thing that you guys can see is if we look in going from 0 to 15 minutes, if you actually measured that, you notice that the spindle's elongated. And that is because the poles are actually moving further apart. So when we get to anaphase, here, two things are happening at the same time that you're going to see, but we can separate them. One event is called anaphase A. And that's describing how the chromosomes move towards the poles. But at the same time, we're going to have another process called anaphase B. And B means while the chromosomes are moving to the poles, the poles are moving further apart. And again, the purpose of having both of these movements is to get everybody out of the middle of the equator where cell division is going to come through and take place. All right? Cool. Now, the top set of panel here, this is what happens if the cells are expressing normal cyclin B. So that's cyclin B that can basically get ubiquitinated and degraded. Look at what happens now. If instead of having normal cyclin, we're expressing a non-degradable cyclin. And the way that you can do that is, again, you go back to the cyclin B gene, and what you can do is make non-conservative substitutions in the destruction box. That's one way you can do it. The other way you do it is you take out the conserved lysine that gets ubiquitinated. Okay? So in either case now, you've got a form of cyclin B that cannot be degraded. That means you're going to go into mitosis, and MPF activity is going to come up. But now you can't lower it. Okay? So what happens here is MPF activity remains high because you cannot degrade the cyclin subunit. Look at the difference, and this is, again, this is the cool thing about mitosis. All you got to do is look at the cool pictures. Look at the difference now. Okay? We start off, we're in metaphase. We start to go into anaphase. It's not moving quite as well, but we're still getting into anaphase. But look what happens at the end. Look at the DNA. The only difference between this cell and that one is that this cell is still expressing high levels of MPF. It's still active. That means the kinase is still phosphorylating things. So among the proteins that are being phosphorylated are the proteins that are involved in condensing your chromosomes. You notice that normally when MPF is degraded, those proteins get dephosphorylated, and that's what causes your chromosomes to decondense. They can't do that here. Okay? So one of the targets of MPF that you can see right here are proteins that are involved in chromosome condensation to basically mobilize your chromosomes to be able to move them. But you notice the cell still goes into anaphase. So even though MPF cannot be degraded, the cells can still go into anaphase, and that's because the anaphase promoting complex was activated. It just means it can't get rid of MPF like it normally does. Okay? So that's what you can come up with this experiment. Now we're going to do a variation of that experiment. Now what we're going to do is to throw in an inhibitor of the anaphase promoting complex. So now instead of just blocking cyclin B degradation, we're going to inhibit the ubiquitin ligase itself. And the way that you do this is that the inhibitor, you notice it doesn't tell you anything here, it just says plus APC inhibitor. The inhibitor is a little peptide. And what it is, it's a little peptide that has the amino acid sequence of a destruction box. It's a decoy. And what it does is that as you put this into cells, and you have to do this as a dosage experiment. So you notice here that what you're doing is that you're increasing the concentration of this little peptide. So if you don't put any of it in, it does inhibit the APC. You put in more and more and more, and what happens is that peptide, it binds to the anaphase promoting complex. It looks just like the destruction box on a cyclin. So the anaphase promoting complex binds to it, but it inhibits it. It keeps it from ubiquitinating its normal substrates. So what you do is you get up to a high enough concentration of the peptide, you're going to completely inhibit the anaphase promoting complex. If you don't put in all that much, it slows down anaphase. Okay? But look at the difference here in this experiment from the previous one. They're not the same outcome. Okay? If we take a look here, so what you're comparing, here's what goes on normally. And what you're having to do is to kind of compare the time points here. They're not identical. So what we're looking at here, here's the 15-minute time point. Okay? That's if you don't do anything. the cells have made it into anaphase. So start there. We're starting with this point up here. Here's the 15-minute time point if we don't add an inhibitor. Cells are in anaphase. Now look what happens here if we throw in a relatively low dose of the drug. Okay? Notice they haven't made it to anaphase yet. We've slowed it down because now instead of being at anaphase like they should be, they're still lined up at metaphase. You've slowed it down. Okay? If you basically go up to a higher concentration then, you can see here, now you can notice that they actually get to anaphase, but it takes them 40 minutes to get there. You've really slowed it down. Okay? So the point is this, and then you go to the highest concentrations here, and now you've totally killed the anaphase promoting complex. So here's what it's showing you. The anaphase promoting complex, number one, if you inhibit it, it can't degrade cyclin B. So what happens in the presence of this inhibitor, MPF activity stays high. Okay? That's why the chromosomes remain condensed. But it's also saying there's another substrate for the anaphase promoting complex besides cyclin B. And that substrate has to be acted on by the anaphase promoting complex for the cell to go into anaphase. And what you're going to see is there's a protein that holds the sister chromatids together all the way up through metaphase. And the anaphase promoting complex has to come and ubiquitinate and cleave that protein, and that's what allows the sister chromatids to separate in anaphase. That's how the anaphase promoting complex got its name. So there's going to be another substrate for ubiquitination and degradation, and that's involved with a protein that holds your sister chromatids together. Okay? So the point is, cyclin B is just one substrate of at least a couple for the anaphase promoting complex. All right? Okay. Here's the first, let's go through now what has to happen. When the cell goes from interphase to prophase, so this is very, very late G2. First thing we have to do is get rid of the nuclear membrane. Okay? We've duplicated the centrosomes. They're good to go. We're ready to make the mitotic spindle, but we have to get rid of the nuclear membrane. The target is one of the intermediate filament proteins, lamin A or lamin B. We haven't talked about them yet, so here they are. The lamins form a mesh under the nuclear membrane. So if you look at an interphase cell, this is staining with a fluorescent antibody against nuclear lamin. And what you can see is that all of the staining is right under the nuclear membrane. And that's because the lamins have polymerized into intermediate filaments. But as the cell goes into prophase, they depolymerize. And that depolymerization is because the lamins are phosphorylated by MPF. So this is the first substrate for MPF. It's going to phosphorylate certain serines or threonines on the lamins. Here's what shows you how dynamic the lamins are. So this is a cell that's expressing GFP tag lamin A. So here's what's happening in real life. This is what your nuclear membrane does every time one of your cells divide. It really vividly shows what's going on. Right there. What's really cool about it is they segregate with your chromosomes. And here's what's fascinating to think about from the structure of your nucleus. The entire nuclear membrane gets dissolved. It literally depolymerizes at once when the cell goes into prophase. But if you notice, when we come out of mitosis and we have to reassemble the nucleus, you do it chromosome by chromosome. You literally reform a membrane around every single one of your individual chromosomes, and then they fuse together to generate the intact nucleus. It's like vesicle fusion, okay? So we get rid of the nuclear membrane all at once. We reform it chromosome by chromosome, but you can see it's incredibly dynamic. Okay? All righty. That's what the nuclear lamins look like. If you were inside your nucleus looking up at the ceiling, this is what you'd see. It's this meshwork of lamins that polymerize to make this crosshatch structure of intermediate filaments. Remember, the nuclear membrane is a phospholipid bilayer. It's pretty flaccid. So the way to think about it is the nuclear lamins are on the nuclear side of that membrane to basically give it integrity and to give it strength. So they're underlying. They're doing exactly the same thing that actin microfilaments do for your plasma membrane. Okay? But they make a barrier. Because if your microtubules on the outside in the cytoplasm trying to get into the nucleus, you can't get past this. So in order for the cell to make that spindle, we dissolve these. And the way that we do it is that if we take a look, here's the building block structure of the lamins to make intermediate filaments. And they come together to make these head-to-head tetramers. Okay? And they basically come together, and that's how they make this lattice work. The lamin A is phosphorylated by m4a. So the serines and threonines that are the phosphorylation targets, you find them clustered at the very amino terminus of the protein and at the very C terminus of the protein. And so what happens is when you phosphorylate the lamins, that's what causes your nuclear membrane to depolymerize. You put it back together again when you come out of mitosis by dephosphorylating the lamins, and they'll spontaneously reassemble. So here's a case of phosphorylation that's being used to directly affect the polymerization and depolymerization of a cytoskeletal component. So, of course, as you guys know, how can we play with this? All right? Well, what happens if you go in to lamin A or lamin B and change all those phosphorylation sites to alanine? We eliminate the ability of the protein to be phosphorylated. And you can predict you're going to screw up mitosis. Okay? So here's what's going on. If we take a look now, we're visualizing chromosomes here as a cell goes from interphase to prophase to metaphase. You look at the chromosomes by staining again using a fluorescent dye that binds DNA. So there's the chromosomes. The same images now, instead of taking a look at microtubules and tubulin, now we're staining with an antibody that recognizes lamin A. Okay? So take a look up here. Here are the chromosomes, and we're only following going into metaphase. Okay? If we take a look over here, here's where you can really see all that intense staining of lamin A, and it's right under the periphery of the nuclear membrane. That's where it's all polymerized. Okay? Look what happens as the cell starts going into prophase. You notice that the intensity starts becoming very diffuse. That's because the lamins are depolymerizing as they're phosphorylated. Okay? The proteins are still there. They're just not polymers anymore. Okay? The other thing, can you guys see, if you look at this, up here you notice that the nuclear membrane is nice and smooth. Look at it at this point here, and you notice it's starting to become wrinkled and puckered. Those are because of microtubules on the outside that are starting to push on it. So they're actually trying to break through and get into the nucleus to form attachments with the chromosomes. Okay? That's at a very early stage, so they're not succeeding. But can you guys see how the nuclear membrane is being distorted? But now when we get to metaphase, that intense staining is gone. The lamin is still there, but it's now completely depolymerized. And as a consequence of that, can you guys see that we were able to successfully make a mitotic spindle? In fact, this is just the earliest point of anaphase where the chromosomes are just starting to move apart. Look what happens if you cannot phosphorylate lamin A. Big difference. Okay? We start off up here. Here's interphase, very intense staining. Look at the chromosomes. we can't form a mitotic spindle. Because if you take a look here now at prophase, you notice we're not getting any diffuse staining. And that's because the lamin cannot depolymerize. The nuclear membrane is intact. But look how its structure is very distorted. It's being pushed all over the place by microtubules that are coming in and trying to attach to the chromosomes, and they can't do it. So even here, you can really see how the nucleus is being squeezed. It's really being impacted by microtubules that are trying to hook up, and they can't do it. And so you cannot force the chromosomes, in this case, to form a mitotic spindle. It's defective. Now, you can only do these experiments under very carefully controlled conditions. You can't express this mutant form of lamin A all of the time because the cells will die. You have to switch it on and switch it off so that you can study what happens. Because what will happen with these cells, if they stay like that, they will activate apoptosis and die because they cannot complete mitosis and can't segregate their chromosomes. Okay? All right. Here's another aspect of lamins. Okay? It's a very, very rare but horrific disease called Hutchinson-Gilford progeria. Okay? HGPS. Some of you may have heard about this. Okay? It's a very rare disease, but what makes it particularly horrific is that this disease causes accelerated aging. Okay? So what it means is these individuals who have HGPS, they die of old age in late childhood, in their early teens, typically when they get to about 12, 13, 14. They die of old age. This disease causes rapid, accelerated aging of every cell and tissue in your body. So it's not just affecting a subset of cells, it's systemic. So you can see from the appearance of these kids, they literally look like old people. They not only look like it, they have all the physiological impairments of people that are in their 70s and 80s. Atherosclerosis, skeletal muscle disorders. Their bones become brittle. They have osteoarthritis, osteoporosis. And it's tragic because the kids die of old age in their teens. Who's the culprit? Lamin. And what makes it really insidious? This is the cause. A single nucleotide transition. A C to T transition. And what it does, it gets even more bizarre. It activates a cryptic splice site. The lamin A gene, like many, many genes, has multiple introns, exons. They all have to be spliced. But by changing this one cytosine to thymine, you activate a cryptic splice site within exon 11. That's part of the protein coding region of lamin A. And what it does is it takes and it splices out and makes a perfect 150 nucleotide deletion, 50 amino acids near the carboxy terminus of the lamin A protein are gone. So everything up to that point looks like lamin A. Everything that comes after that looks like lamin A. It's missing 50 amino acids near the carboxy terminus. That single point mutation causes this accelerated aging. Okay? One point mutation, you alter a splice site, one protein, and you suffer the consequences of premature aging. Okay? The protein is now referred to, this mutant protein, by the way, you'll see it referred to as progerin. It's a mutant form of lamin A, but we call it progerin because it's the causative agent of progeria. Okay? If you take a look at what treatments are out there for progeria, it's really limited because the aging starts as soon as the child's born. Okay? So as soon as you emerge, you're already aging. So the therapeutic options that you have there is there any way that you can correct this lesion? Okay? Well, the technology that's probably ultimately the only one that's going to work is to do gene editing. And that would require doing gene correction or gene editing on a pre-implantation embryo that's going to have that particular point mutation. And literally going in and probably using CRISPR-Cas technology to go in and change that T back to a C. That is ultimately the only way you can prevent the disease. Anything you do after that is to ameliorate or to retard the rate of aging. But you can't stop it, and the problem is, as you guys know, we can't reverse it. So any of these other therapeutic options that you can look at here, you're trying to just stave off the inevitable, okay? Now that's not to say that you can't do it, but the point is this, is that any of the therapies that you're going to be looking at, including the one we're going to look at here in a second, all you're doing is you're trying to slow it down. You're trying to stave off the advanced aging and trying to at least give these individuals something of a little bit more expanded longevity by trying to treat it. Okay? So here's what this mutation does. We knock out 50 amino acids near the carboxy terminus of lamin A. Well, remember we talked about, up there early on, we talked about lipid-anchored proteins. It was another way to take a protein and attach it covalently to a lipid so it could be found in one leaflet of the plasma membrane. For example, all the monomeric G proteins we've talked about, they all have lipid anchors. And that's what anchors them and associates them with the cytosolic leaflet of the plasma membrane. Lamin A has to go through this as part of its normal maturation. Okay? So here's how the process occurs normally. If you're looking at a wild-type lamin A gene, the last 4 amino acids of the precursor, so we call it pre-lamin A, the last 4 amino acids are cysteine, serine, isoleucine, methionine. It's kind of bizarre. You've got a protein that ends with the same amino acid it begins with. In fact, it's about the only example out there where you have a protein that both begins and ends with methionine. That's the key thing. Because what happens during the maturation of lamin A, to get it up here into the plasma, into the nuclear membrane, right under the bilayer, is that what we have to do is that we have to covalently add a farnesyl lipid. The lipid is covalently attached to the cysteine. That's the amino acid that's also going to be lipid anchored, or the lipid is added to, for all the G proteins that we've talked about so far. Rho, RAC, CDC42, GS alpha, RAS when we get to it. These lipids are always attached to a conserved cysteine that's near the carboxy terminus of the protein. So we add the lipid anchor. Well, now in the case of lamin A, we have to take it off. And the way that we take it off is that the next step is that these 3 amino acids, the serine, isoleucine, methionine, SIM, gets cleaved off by a peptidase. Gone. Now what we do is that there's a modification by putting a methyl group on the cysteine. So we modify the cysteine. And only after you've gone through all those steps can we now cleave this part of the protein here, the last 15 amino acids, that includes the lipid. So if you look at this, does this strike you as having any purpose? You attach a lipid to a protein only to have to take it off in order for the protein to function. The reason you have to go through this is that this is the way that lamin A gets up, and it's basically held in place by sticking that lipid anchor into the nuclear membrane to hold it there while it gets processed. It's just kind of a mechanism that the cell uses to hold the protein in place to get it up near the nuclear membrane where it's going to have to stay. Cool with that. look what happens with progerin. Here's where the problem comes. These are the amino acids here that are deleted. Those 50 amino acids are gone because of that splice. So basically, when you're splicing the pre-mRNA, these sequences are excluded. So we go right from this amino acid up to the very end with the CSIN. First step occurs naturally. We put the lipid anchor on. Okay? Second step occurs naturally. We cleave it off. Everything looks good with the peptide. But now we're missing these amino acids here. And those 50 amino acids are necessary to cause this last cleavage of the terminal 15 amino acids with the farnesylate. So this horrific disease is because we have a farnesyl group. We have a lipid anchor covalently attached to the protein that we cannot remove. It stays there. So now what happens? Your cells are accumulating and expressing progerin. The protein has a lipid anchor. This stuff is now going to start sticking itself into the nuclear membrane. Remember, the nuclear membrane is also contiguous with what other membrane? The endoplasmic reticulum. So what happens is that over time, all of the lipid membranes within your cell start filling up with this lipid-anchored lamin A, the progerin. And it basically ultimately perturbs every process in the cell, because every single one of your membranes has this protein in it that normally shouldn't be there. And it literally compromises every physiological process in the cell and ultimately causes them to go senescent and to die. Okay? So now you're looking at this and you're going, okay, short of having to do gene editing or gene therapy, is there a pharmacological fix here? What do you think? What would be the most effective way you could keep this disease from happening pharmacologically? Not keep it from happening. How could you treat it? If you could prevent the lipid from being attached in the first place. So the question is there an inhibitor out there that would block the enzyme that covalently attaches the farnesyl? It would be a farnesyl transferase inhibitor, or an FTTase inhibitor. Sure enough, there's one out there. Okay? The scientist who leads all of this work, by the way, Leslie Gordon, her son was diagnosed. Her son had progeria. So she has spent literally her entire career trying to work on this disease because her own son died of it. Okay? Here's what she found. There's a drug that was out there that was developed as a chemotherapeutic drug. It's this drug up here called lonafarnib. Okay? As you're going to see next week, lonafarnib was developed to target the oncogene RAS. RAS is a lipid-anchored protein. So an attractive chemotherapeutic target would be, in order for oncogenic RAS to promote abnormal cell proliferation, it has to be lipid-anchored. So if you could design a drug that prevents RAS from being anchored to the plasma membrane, you can't keep it from being an oncogene, but you can prevent it from causing cells to proliferate. That was the premise for lonafarnib. It failed as a chemotherapeutic drug, and you'll see why next week. And that's because cancer cells are remarkably insidious. And that is, if you prevent a lipid anchor from being attached one way, they'll just use a backup and put it on a different way. They didn't know it at the time. So it turns out this drug, lonafarnib, was the only drug out there that could be used to target the farnesyl transferase. It was a failed chemotherapeutic drug, but at least it's a relatively potent inhibitor of farnesyl transferase. So what they did is they took the drug and initiated clinical trials to use it to treat kids that have progeria. So it's the first drug to be used in a clinical trial to try and ameliorate the effects of progeria. And it looks like it might be working. So this is just the abstract in the paper that actually, as you can see, it came from Gordon's lab. And in essence, what they're doing now is that this was the first report of using lenarfinib to basically block the farnesyl transferase by inhibiting it. And if you go through what you see by looking at the abstract, it doesn't stop the disease, but it at least retards and ameliorates its progression. It slows it down. Okay? Now, is this necessarily going to provide a normal lifespan for these kids? No. But what you're basically just trying to do is to slow down and retard the process of the aging, because it's basically all we can do at this time. So it's interesting that you have a drug that was developed to stop abnormal cell proliferation. And what it is being used for now clinically is to try and stop abnormal cell senescence, the exact opposite physiological effect that it was initially intended for. So it's another illustration of drug repurposing. You take a drug that was designed to basically treat rapid proliferation, and now what you're trying to do is you're using it to try and prevent cellular senescence. Okay? So just a little story there that illustrates the importance of covalent lipid attachments to basically during normal protein biosynthesis. Okay. That was all of that, by the way, just before you forget, all of that is to get rid of the nuclear membrane so your cells can divide. Okay? First target of MPF. Here's the second one. MPF has to take your DNA that's strung out in cells and condense it into these very, very, very tight, compact mitotic chromosomes. In essence, you have to take DNA that's already balled up, and it's already pretty compacted in your cells. Do you know how long the length of DNA would be if you popped open one of your cells and spread it out? It's about 3 feet in length. That's how much it's already compressed to get it to fit inside of a cell. In order to take that DNA and to pack it even more for mitosis, the cell's got to use everything it has to compress it even further. The starting point is this. As you guys all know, the starting order to take DNA and put it into chromosomes is we wrap our DNA around structures that are called nucleosomes. And nucleosomes consist of octamers of the core histones H2A, H2B, H3, and H4. And it's basically called the beads on a string or the solenoid model. But what it does is you wrap your DNA around nucleosomes that are adjacent to each other, and you've got about 200 base pairs of DNA inside each nucleosome. Okay? Now, if you take a look at that, what limits being able to compress it even further is that the histones are the most basic proteins in the cell. They're loaded with lysine and arginine. They're incredibly positively charged. That's how the DNA wraps around them. It's all electrostatic. So what keeps these nucleosome octamers from getting closer together is that the positive charge on the histones flings them apart. So one of the ways that you can basically pack histones together more closely is to phosphorylate a little protein that's here in what's called the linker region, in between them. And that's the linker protein histone H1. It's in between, it's not shown here on this figure, but there'd be a histone H1 in between each of those octamers. Well, when you phosphorylate it, you give it a negative charge. That neutralizes the electrostatic charge repulsion of adjacent nucleosomes, and they're actually attracted now to the negative charge on histone H1. So in essence, if we put a negative charge in here with histone H1, it allows these guys to come closer together. That's a substrate for MPF. It's a start, but it's not the only one. We come up here now, and there's another protein that takes chromosomes and packs them even tighter. and it's a protein that's called condensin. Name tells you what it does. So what MPF does, it also phosphorylates condensin. So you start here, you phosphorylate H1, you phosphorylate condensin, and basically now you pack all of that DNA into very, very tight structures. So now you guys can see, if you have non-degradable cycling, and you cannot get rid of MPF activity during mitosis, you can't reverse the phosphorylation of these proteins here. So that's why the chromosomes in that first set of slides remain condensed after the cell completed mitosis because the kinase is still active. So in order for a cell to go back into interphase, we basically have to dephosphorylate histone H1 and condensin in order to get the chromosomes to decondense and to go back to interphase. Okay. Here's the protein, just to finish up. Here's the protein that holds your chromosomes together. It's a protein complex that are called cohesins. Cohesins have to hold the sister chromatids together after DNA replication has taken place. So all I want to show you here are two things. Here's a chromosome, okay? Double-stranded DNA. As we go into S phase, or before, in G1, we start assembling cohesins along the length of the chromosome. Now we go into S phase. We have to separate those two DNA strands, and now what we do is we semi-conservatively replicate each one. So now what we're generating are what are called sister chromatids. All right? We got one here, one here. And what you can see is as the replication fork comes through, and it'd be moving in this direction, we're now generating two new DNA double helices. We have to hold those sister chromatids together, and that's what the cohesins do. So as DNA replication proceeds, our two sets of sister chromatids are held together by the protein cohesin. Okay? What's going to be loaded at the centromere, and we'll start off with this on Monday, is that as we load off right here, we're going to recruit a series of proteins that go into the kinetochore. The kinetochore is going to be the binding site for microtubules to attach to the chromosomes. There's going to be a kinase in there and a phosphatase in there. And what we're going to do is we're going to regulate how tense these two sister chromatids are applied to by the mitotic spindle by proteins right here being moved to where they're either phosphorylated or they're going to be dephosphorylated. The basis of going into anaphase is protein phosphorylation and dephosphorylation. And what's holding it all together right here is this small number of cohesins. That's what's holding your two sister chromatids together. The proteins that we have to get rid of in order to go into anaphase and send this sister chromatid this way and this one over to the right is that the anaphase promoting complex has to cleave the cohesins. And that's what allows them to separate and we can go into anaphase. Okay? All right, that's where we'll start it off on Monday. I've got the exam scores. I'll get them posted in about 10 minutes. Okay? Have a good weekend.